Yeah, it's my personal evolution about optimum pH. And I now recommend, well, we do, but I recommend higher optimums, mainly because I think they're safer for nutrition. You know, if the pH gets, it's kind of like where do you drive in a road? And if there's nobody coming, drive right in the middle of the frickin' road. It gives you the biggest buffer on both sides. And pH, if it should drift low, you get micronutrient toxicities. If it drifts high, you get iron deficiency, except in cannabis. Cannabis just gets iron all the time. So we've been using a higher pH for cannabis to make sure we don't get toxic uptake of especially manganese.
Now, your recommended pH was 5.5 to 5.8, but with the cannabis plant, what are you guys running right now in the lab? And
more like 6.5.
6.5?
6.5, yeah.
And on commercial scale cultivation regarding pH, and what kind of media are you in?
Lots of them,
Yeah.
because we're in research. But our standard media that we've used forever and ever is a peat-based media. And we could get into how we evolved exactly what's in there, but the main ingredient is peat, Canadian peat. But we've also grown a lot in coconut coir. We've also grown a lot in mineral wool, like Grodan, and then a lot of studies in straight liquid hydroponics, too.
Now, the recommended 6.5 pH, you're kind of testing that in all medias, or is there specific medias that you're running that in?
No, in all media.
Okay.
And now, this is one of those things that's more of a good sounding hypothesis than that we have a ton of research data for. But we have had instances more than once where the pH accidentally drifted low and we got manganese toxicity and the black spots on the leaves. It's a classic symptom. And they test high in manganese. And it's really bad, and then you're trying to get the pH back up and everything. In other crops, if we grew them at 6.5, we would start to get iron deficiency chlorosis, interveinal yellowing between the leaves. That's really bad, and you have chelates to prevent that and to minimize that. But man, cannabis, you could have iron 5 counties away and it'd find it. I mean, it just never has iron deficiencies. So we can now err on the high side for pH and minimize the potential for micronutrient toxicity.
That'll be interesting. We should run, 6.5 pH on some side-by-sides. But is it really necessary, though, since we're not having any manganese toxicity? So shouldn't we just keep doing what we're doing?
Sure. You should. There's nothing wrong with it. If you haven't had a pH-controlled failure on the low side, it's manganese is unique among the micronutrients. I may be one of the few people that keeps saying manganese has active uptake, which means the plants just suck it up fast. You give them a big reservoir and they'll just draw the manganese down to 0. They take it all up. And no other micronutrient is like that. And so you got to be careful about not having too much manganese. The most specific thing that happens, and now this is a little bit of chemistry, but all these micronutrients form hydroxides, MnOH. And OH is a hydroxide. And OH is what makes water alkaline. And MnOH is not soluble and doesn't go through plant membranes. So when the pH is high, you get manganese hydroxide, and the plant can't take that up. And it goes low, and all those manganese hydroxides solubilize, and you got pure Mn divalent cation in the solution. So it's all because of the chemistry of the solution. It's better to say blocks the uptake would be something like at the membrane level. It's better to say it makes it in less bioavailable. Technically, that's what a chemist would say. As you mentioned, a probe fundamentally measures the electrical conductivity of the solution. It doesn't measure parts per million. It's a calculated number. It measures electrical conductivity.
Yeah, for us, the reason why we wanted to kind of push that movement was communication. When it's dealing with parts per million, you have two different measurements, 500 and 700. And when we're communicating via DM or message and understanding that EC is the universal language, it's better to communicate in EC. Bruce, I'm always fascinated on what people are passionate about and how they got into their fields. What age did you develop a passion for plants?
Yeah, it was later in life. I mean, my parents didn't really have a garden, and we lived in a rural area, but we weren't farmers. Unlike you, I know that you got interested in kindergarten. But for me, it was taking a botany class in college and germinating seeds, and just boom, it was like the switch went on. And I was in engineering at the time as a student. I guess I could say I found the other engineers a little stiff about their approach to life. And I didn't fit in with that as well. But so I switched to plant sciences and botany and liberal arts. And I started growing a garden, just getting really involved with plants. Then it went from there. I started to take classes in school, and I didn't really know what I was going to do as a profession. And it turns out I never had to figure that out. I just kept studying and taking more classes, and pretty soon you end up being a professor at a university. And it's like, I never had a real job. I just kept
doing it. What do you love about horticulture?
I think it's the caring for another living organism. People care for their pets. They care for other people. That's part of biology. And in this case, it's taking an organism and how fast can you make it grow? That's pretty interesting if the conditions are optimal. And if something goes wrong, then how do you fix it? And what is it that went wrong, and how do you fix that? For me, that's a pretty fundamental passion. And I think that would probably explain a lot of other people, too, the reason for getting into it. I got to tell you a little story about this. So we get lots of funding from NASA my whole career. And NASA has a greenhouse at the South Pole. And that greenhouse is in complete darkness. I think it's 90 days a year. The sun never rises. And so you would think the people at the South Pole would just be in that greenhouse all the time, because they grow tomatoes and lettuce to eat. And then we do studies. We means NASA does them, but I see the results. It turns out there's a significant fraction of people that say, "No, I'm fine without plants. I don't need to go to the greenhouse." And the other people say, "I go there every day. I sit in the greenhouse and read a book." And I find that just fascinating. And I always wanted to follow that up. The people that don't think they need plants, what else is going on in their lives? I mean, I bet they don't have pets either. Stuff like that. And then the question comes up, okay, if you're going to go with NASA on a trip to Mars, who would you like to go with? And I said once in an interview, well, those people that don't like plants, I don't want to go with them. I get nervous.
Did you start off at another university, or you always went to Utah State?
No, no one would keep me very long. I grew up in Minnesota, rural Minnesota, and I went to the University of Minnesota as a freshman, and I graduated from there. And then here we are in California, but I'd been fascinated by just life in California. The free speech movement at Berkeley, and at the time I had a girlfriend that was at Berkeley, and off I go to California. And I started a master's degree at UC Davis. We won't talk about what year that was, but it was a while ago. And that was great. That was a great school, and I was in the Department of Vegetable Crops there, so really focused on vegetables. And then an opportunity to work on a Ph.D. came along at Penn State University, so other part of the nation, but also a very big agriculture school. And that combined my engineering background with plants because we were working on energy efficiency in greenhouses, Rootzone heating and optimal temperatures. And then at the end of that, a position opened up at Utah State University. And there was a famous plant physiologist there named Frank Salisbury that wrote the best-selling plant physiology book ever written. And it was an opportunity to work with him that brought me back west to Utah. And then the rest is history, right? I've been at Utah a long time now.
Now, when did you guys start the cannabis research program at Utah State?
Right after it became federally legal to grow low THC cannabis. There were some growers in states where it was legal that wanted to fund us to do research, because we've been doing research on indoor agriculture with NASA for a long time, nutrition, lighting, all the same things. And I knew these big growers, and they said, we would like to fund you. And the university, up until that time, said, no, we can't take this money. Too risky. We'll get crossways with the federal government, and you can't do cannabis research on campus. It had to be sort of clandestine. But then, gosh, what year was that? 2018? I've forgotten the year.
Yeah, when the DEA legalized hemp.
Yeah. And the legal counsel said, okay, it's safe now. You can take the money and start doing research. So that's when it started. December 18. It was December, and I think it was 2018. And we knew it was coming. And we have something like 44 growth chambers in my lab. I mean, we were ready to go right out of the gate with cannabis research.
Yeah, because you were focused on indoor cultivation, being with NASA, and so you went right in. You were ready.
Yes. We've done very little field research, a little bit in collaboration with my colleagues in the field, but it's predominantly greenhouses and growth chambers.
How many cannabis research facilities are there in universities across the United States?
I think on paper, well, first of all, there's one agriculture school in each state. And here in California, it's the UC Davis. On paper, all of those schools have some kind of a cannabis program. Almost all of them call it hemp, because that's a safer sounding word. And nobody calls it marijuana.
Yeah.
But they call it hemp. And most of them are studying it for fiber and for feed, for seed for animals and seed for people. It's much more unusual to have a medical cannabis research program, but there are a few schools, maybe even several now schools that are doing medical cannabis. So it's not totally unique. I think maybe the difference is they didn't all have the infrastructure to get ramped up as fast as we did.
Yeah, it was when I came over and visited your lab. Back then, I knew you were huge in lighting, and that's why we worked together a lot on what the cannabis plant utilizes for lighting. But then going over and visiting your lab, I realized, wow, you do way more with nutrition. I mean, it's why do you think that is? You think, more of YouTube and the channel is more about lighting, or?
Yeah, it's a good question. I mean, part of it probably has to do with my company, Apogee Instruments, which sells instruments to measure lighting, quantum meters, spectroradiometers, all that kind of stuff. And so that's helped. We do a lot of, we call it photobiology research, and we did a lot of that with NASA too. What ratios of colors, how can we manipulate plant growth with that? And I think it's really because people are more fascinated with colors of light in plant growth. You can see the colors and what should we do? Nutrition, you got a bottle of nutrients and it looks the same as one that's completely different. That for sure affects plant growth, but it's not as a direct effect as lighting. And lighting, of course, commercially is huge because now all the lighting companies are making all kinds of lights. So I think what I'm trying to say is it's just such a visual effect to do lighting research.
Yeah, I looked at one of your papers you published, Principles of Nutrient Management for Closed Systems. And can you talk about the goal with that study?
Yeah, that was actually sort of a review paper, although we have some new data in that as well. But in a nutshell, it was driven by our research for NASA. And NASA cannot have runoff. They can't have leaching. You just put the water in, you put the nutrients in, and it has to stay balanced week after week, month after month. You can't fix your mistakes by leaching it out. There's nowhere to go. So what's the ratio of nutrients and water that you would put in to keep the plant optimally healthy with no leaching? And I think you've often called it runoff too. 0. So it was driven by that. And what we have done is use this principle called mass balance, and the nutrients have to go somewhere. You put them in the water, and initially they're in the water, not the plant, and then they go in the plant, and now how much do we want in the plant? And let's say we want nitrogen, we want 4% nitrogen in the leaves. Well, how much do we have to put in the water to get 4% in the leaves? And you go through all of the elements that way, and you can choose the number you want for the leaves. Let's say we want 0.4% phosphorus. How much do we put in the solution to get 0.4% in the leaves?
Now, what is the definition of mass balance?
It's related to energy balance. Mass has to go somewhere, can't disappear. It has to, at the end of the study, if you put 100 grams of, say, nitrogen in there, you're going to have 100 grams somewhere in the system.
Somewhere in the plant.
It's either going to be in the plant or the solution.
Now, are you seeing, now knowing that you can't have any runoff or the right technical term is leaching, are you seeing different fertilizers get picked up in different amounts? And how do you control that?
They definitely get picked up in different amounts. The 3 big elements are nitrogen, phosphorus, and potassium, NPK. And because of that, all fertilizer bags have 3 numbers, and that's the nitrogen, phosphorus, potassium in the fertilizer. All 3 of those have active uptake. So if you take a plant in a container and you put the fertilizer in, and you start measuring the nutrients. Let's say we measured them every hour. We got plenty of money. We're just measuring them all the time. All of the other elements would stay relatively constant, but the N, the P, and the K would have active uptake, and the plant will draw those to near 0 in the solution. And then you measure it, and you go, oh my God, there's 0. We need to put more in. And you put more in, and the plant takes those up, and you more in, and the plant takes those up, and pretty soon you have a toxicity problem. And mass balance says, no, you've put those in the solution. They're in the plant where we want them. Don't worry about it. If it's low in the solution, that's a good thing. That's a healthy plant. It took them out of the solution. don't panic and keep putting them in because you've already put them in. The analogy I've used is like feeding a dog. You feed a dog, and in 1 minute it'll eat all its food, right? And the bowl is empty. The dog must be hungry, and if you keep doing that, you get such a fat dog. You roll over and put its paws in the air. But you know that the dog had plenty to eat, and if the bowl is empty, you fed it, and that's fine. So that's the same principle as fertilization, where we don't fundamentally go by the concentration left in the media. We go by what we put into the media.
Are you seeing ratios come? How are you feeding with different ratios of fertilizer at different times, or it's always the same inputs?
Yeah, that's a really good question. Let me address it in a big picture. But was it Socrates that said the unexamined life is not worth living? Well, I kind of twist that and I say the unstressed life is not worth living either.
Yeah.
We give ourselves stress, and that's what gets us excited about things. And the same thing with plants. An optimal plant is not completely free of stress. You get a big lush plant that it would get diseases more quickly. So the optimum plant has precision stress. And one of the key ways we can stress it is nutrients. Get those at just the right levels to make the plant have to work harder to do things. And there's two approaches to that. One is keep the nutrients low to make the plant work hard to get the nutrients. And the other is to have them high so the plant works to keep toxic levels out. And both of those are used as a way of precision stress. As a first pass, the ones that have active uptake are also mobile, meaning they can be redistributed in the plant. And being mobile in the plant's a very good thing. It moves it where the plant needs it the most. But interesting, NPK are the three elements that are the most highly mobile. And what that means is that older leaves get deficiencies first because the plant cannibalizes them. But it's not completely true because manganese has active uptake, and that is minimally mobile in the plant. It goes in the old leaves and it stays there. We've had lettuce plants that we test the old leaves, the medium leaves, the young leaves. The old leaves have what would be considered toxic levels of manganese. The middle leaves are right on the money, and the young leaves are deficient. And it's the same element growing in the same solution, just because it's not mobile. Calcium for sure is like that. It goes in the old leaves, and they just go higher and higher and higher. And here's these young leaves they have very low levels of calcium because it's so immobile. But back to the short answer. Yes, if they have active uptake, they're typically always mobile, but there's a few exceptions. Yeah.
Can you explain what WUE is?
Yeah, in the case of plants, it's water use efficiency. And the units are grams or pounds of plant biomass per volume of water transpired. And that could be pounds per gallon. In our case, it's usually grams of dry biomass per liter of water. And it's a critical ratio because that affects how much fertilizer you have to put on. If the plant's going through a lot of water, you water a lot and the fertilizer can be very dilute. And conversely, if it's in like a high humidity environment, hardly going through any water, the fertilizer needs to be much more concentrated. And so what changes water use efficiency? In the greenhouse, with lower humidity and sunlight, the water use efficiency is typically about 3 grams of biomass per liter. Now we take those plants and put them indoors. Humidity is high. Carbon dioxide's typically elevated, and that closes stomates, makes the plants grow faster. And they're under LEDs, which are cool. There's not such a big thermal load on the plants. So that water use efficiency can go from 3 to as high as 6 grams per liter. And what that means is we should be doubling the concentration of nutrients that we're putting on because they're growing fast and they're not using very much water.
So when we're needing to feed a higher EC value with CO2, it's not that the plant's working harder, it's that the water use efficiency
is better, is higher. Yeah.
And the same, because I was always under the assumption that higher PPFD with LED lighting is increasing energy to the canopy, which is making us feed a higher EC. Is that the case, or?
Partially. If the technology changes from high pressure sodium to LEDs at the exact same light level, the heat from the LED lights would make them use more water.
Or HPS, you mean?
HPS, sorry.
Yeah,
Did I get that backwards? Thanks. HPS. The heat from the HPS makes them use more water. So you would make it more dilute. You move to LEDs, they're using less water. It needs to be more concentrated. So that's a fundamental principle, and we've shown that. But what if you took a PPFD of 500 and you raised it to 1,000? You doubled it, you'd think, whoa, these plants are growing a lot faster. But in that case, the water uptake also doubles. So it's the same fixture, same room. So the ratio of water and nutrients doesn't change with light level. You can double the light and you end up doubling the growth rate and you double the transpiration and the water use efficiency stays the same.
So it's more about transpiration.
Mm-hmm.
Now, in regards to CO2, why does that affect water use efficiency so much?
The way I like to explain it, first of all, the word stomata comes from the Greek word for mouth. And so here's us. When we run, you're wide open, you got to have more oxygen. You're also breathing hard, you're losing more water. Well, plants growing faster, it's got to open its stomates to get carbon dioxide. And as soon as it opens its stomates, the water starts to go out faster. So this is a big challenge for all plants, how to get CO2 in without losing too much water. And plants have, boy, that is very well studied in the plant physiology literature. But now, using this analogy with people, what if we said, we want to make it easy on the people, we're just going to fill the room with oxygen. They don't have to breathe as hard. And we wouldn't have to breathe as hard, and we wouldn't lose as much water because we get pure oxygen. Well, with plants, we give them high CO2. Hallelujah, they get a lot of CO2. Their stomates just close. Why would I have to open my stomates? There's plenty of CO2.
Which automatically changes water use ratio, efficiency.
Yeah. It's really interesting. The numerator goes up because of CO2, and the denominator, water, goes down. And that's where we go from like 3 grams per liter to up as high as 6.
So the plants are drinking less, but you need to feed a higher EC to get the minerals into the plant.
Yeah.
So, I mean, when you're seeing deficiencies, when you say, you weren't running CO2, your CO2 levels are averaging around 500, you come in and you put, add 700 ppms of CO2, bring up the CO2 levels to 1200, and you start to see deficiencies, it's water use efficiency.
Yeah, it could be lack of nutrients.
Yeah, lack of nutrients.
Yep.
Yeah, raise the EC.
Yeah.
And same thing with LEDs because of the heat. HPS has a lot of heat down on the canopy, changing the water use efficiency, and then less heat. I didn't know what water use efficiency was until we talked, but I thought it was really interesting.
It's really underappreciated.
It is.
In terms of nutrition. And that paper that you referenced, that's a big part of that paper, is explaining that to people. And it's a scientific paper, but we have a lot of diagrams in there and figures. And, I hope it's easy to read for anybody.
Talking about CO2, do you need to increase PPFD on lighting when you add CO2?
Not necessarily. It comes down to an economics thing. It's just always cheaper to raise the CO2 up to an optimal level and then put as much light as you can on, depending on the electric bill and the economics of the input of electricity and the value of the crop. But CO2 is a relatively cheap input.
How significant is CO2 regarding quality or, yield on cannabis?
Most of the research on this comes from other crops in the field, all of our major agronomic crops. And we typically say raising CO2 from 400, which is ambient, to 1,200, which, first of all, 1,200 is you're 99% of the way there to saturation of the CO2 effect. There's not economic value of going higher than that. Okay, so we go from 400 to 1,200. That's typically associated with about a 30% increase in yield. That's huge.
And that's without increasing
Variable.
any other variable.
Nothing else changes.
But obviously you're going to have to increase EC just a bit because
Yes.
of water use efficiency. That's significant.
In a lot of cases, we get away with a certain amount of sloppiness in fertilization because we overwater and we overfertilize, and you just wash out your mistakes. But the goal is not to wash out mistakes. It's not to have any mistakes in the first place. Have optimal nutrition so you're not running water down the drain.
Yeah. 30%, it's different coming from you other than like a nutrient company trying to sell a fancy bottle. Like 30, everyone throws around 30%, but to say you actually have studies that are shown 30% increase when using CO2, everybody should be running CO2.
Oh, they should, yes.
That's big. Bruce, how familiar are you with hop latent viroid?
Well, I know it's a big problem in cannabis, and because of that, we've worked with Toomey Genomics in Colorado to test our lines. I know people are. It's showing up everywhere. I know that. So, but I'm not a plant pathologist, so if you get deep into the viral pathology, you're going to. I'm not going to give you detailed answers, but I sure know it's a huge problem in cannabis.
Have you ever thought about doing work on hop latent viroid?
Well, one of the interesting things, first of all, we're pretty sure it came from hops. That's why it's called hop latent viroid. And in the case of hops, all the hop plants have it, but they don't show symptoms. They just live with this virus, and it's not a problem. So it made the jump from hops to cannabis. And in cannabis, it can be what we call asymptomatic, just no symptoms until the plants get stressed. And then you get some big problems. So in our studies, at least the lines we have lines that have it and ones that don't. And the ones that have it haven't shown symptoms. So it's really interesting. Lots of plants show symptoms, but not all. And I'm looking forward to doing more research on that. But it's not a good thing. We don't want it in our plants.
Have you heard of any studies that have cured or cleaned hop latent viroid or any viruses out of plants?
Well, there are lots of studies on cleaning viruses out of plants. They're just not in cannabis. And this is where, I guess as academics, you study other plants and then you take what you can apply to cannabis. But a really good example is potatoes. They're vegetatively propagated, and potatoes get viruses, and they're bad. So we use tissue culture to micro-propagation to take those cells that don't have the virus in them and propagate them in tissue culture to get a virus-free potato plant. And then you regenerate the plant, and you're careful. And I think in the potato industry, I'm going to take a guess, maybe 6 generations that they grow them, and then they say, okay, too risky. We're going to start over with tissue cultured plants that we know are clean. So tissue culture is widely used to get rid of viruses in plants.
Do you think it's important that it's Meristem tissue culture, or?
Well, our understanding of that is because the virus is in all the cells, but if we take just a few cells right in the meristem, they're growing so fast that they outgrow the virus. Those don't have virus in them, and you regenerate the whole plant. So it's really micro-propagation. What if you took a cutting? And you say, well, I'll just take a small cutting. That's not good enough. It just has to be very tip of the plant, which requires tissue culture. And even then, in other crops, it's not 100%. Sometimes you get some cells that's there. You just have to throw those away.
It's really important, being that these cultivators need to always have new, clean genetics consistently, and managing that book of genetics without a lot of labor and a lot of square footage. And tissue culture gives you that ability. I mean, when one of the prototypes I sent out to my place immediately as soon as I got the media from Mike, and all the guys were just running tissue culture right there in the hallway. So it's definitely needed for sure. I know you've done a lot of studies on stressing the root zone in early flower. What have you been seeing with root zone stress in early flower?
The biggest thing is high nutrient stress, osmotic stress, which is like water stress. And the goal is to keep the plant compact. If it's, like we said before, if everything's perfect, the water is high, that you just get a big plant that's not tough. So the goal is to have some high electrical conductivity stress, especially during those first 4 weeks of flowering. Right after the first 4 weeks after the start of short days, when they rapidly elongate, keep the plant compact during that period of time. Now, this is precision stress. So we've done some studies where the EC was a little high, and we got compact plants, yes, but the yield went down 20%. The cannabinoids did not. They stayed the same, interestingly enough. So we keep tweaking this, and generally it's high EC. I think you guys use that too, high EC during the first 4 weeks, and then bring the EC back down.
Have you found a happy medium on stacking EC?
I think the best answer to that is not yet. No. Yeah, as a general rule, first of all, we use pretty dilute fertilizer, so it might be 2 in the beginning, and then we raise it up to 6 and even 8, and then bring it back down. And I can tell you, and I'm sure you've seen it too, cannabis is just tough as nails. I mean, these high ECs, other lesser plants would be wilting. They'd be, oh my God, they're near death.
Now, was it on your 6 to 8 EC that you were seeing the negative effect on yield? Was that the runoff EC, or was that peak EC with dryback?
First of all, in those studies, we didn't have any dryback. We used electromagnetic probe. Mm-hmm. We use probes from Arroyo, they're called now, the Arroyo probes. And so they gave us lots of probes for this research. Incidentally, they just came out with a new probe for this work, which they sent us a bunch of prototypes to test. But we were using that right in the middle of the Grodan block. And then we're simultaneously overwatering just a little bit so we could check the true solution EC. And as a general rule, they're the same. But as we got later in the life cycle, the solution EC was 30 or 40% higher than measured by the probe. And it's like, ooh, wait a minute, is the probe wrong? No, it's because the probe is measuring the EC in the middle of the block, and what came out the bottom was just the last centimeter of the block.
So
doesn't affect the
you're
result.
at like 6 AC full saturation. Full saturation at 7 or 6 or 7 or 6 or 8 EC is, I mean, at 40% dry back, what do you think, like what, 18, 20?
When you do dry back, the salts stay the same, but the water is less. So it can easily double the EC.
So we're not far off in how we run EC in the beginning of flower. I mean, we're probably peaking out around an 8 EC with 40% dry back. So kind of would be about a 4 EC in the block or 3.5, 4 at full saturation.
That's
We usually
stress.
run about a 6 to an 8 EC at 40% dry back.
It's tricky. I mean, we've done multiple studies, and sometimes we see no detrimental effect on yield. But I can't tell you that the EC was perfectly controlled. You know, sometimes we're trying to bring it down, and it didn't come all the way back down.
Bruce, can you talk about some of the studies that you've done on phosphorus?
Yeah, we just published a referee journal article on phosphorus in cannabis. And cannabis it's one of the elements that's unique in cannabis. Typically, crops, we would keep the phosphorus input at 15 parts per million. Even less than that, 12 to 15 ppm. And with cannabis, that low level of phosphorus is fine for vegetative growth. They don't need any more. But then they start to flower, and the flowers don't have any seeds in them with the female plants. And plants are programmed to move phosphorus into their seeds. All plants do this. They give the seeds a good start in life, get them high phosphorus in the seeds. Well, seeds are pretty little, and here's this cannabis with this big clump of tissue up there. There's no seeds, but the plant cannibalizes phosphorus from its leaves and moves it into the flower. That's a mobile element. And so we've seen that by week tests. The phosphorus in the leaves goes from ample to just adequate to below adequate in the leaves. And meanwhile, the phosphorus in the flower clumps is building up like crazy. So you could say, well, if it's building up, the plant must need it. But we're quite certain that all that phosphorus in the flowers is storage phosphorus. There's a compound called phytic acid that just stores phosphorus for future use. It's not functional. So it looks like the plant is out of phosphorus, and we end up giving it more phosphorus. And so how much phosphorus do we need? And our most common thing, we must have done 6 studies on phosphorus, and they're often 30 ppm, 60 ppm, and 90, those 3 levels. And we typically see no effect on yield at all from that. What we do see is an increase in runoff and leaching of phosphorus. And our paper is on sustainable cannabis nutrition. We're trying to get people to use less phosphorus so we don't pollute the lakes and rivers with phosphorus. They get too much phosphorus and they could get algal blooms and toxicities. Phosphorus is a bad environmental pollutant. As much as we are campaigning to get people to use less phosphorus, there is some data from Nurit Bernstein's lab in Israel that shows a careful study on phosphorus, and her high THC cultivar showed no effect. 30, 60, 90, there was no value of high phosphorus. But she had a low THC cultivar that did show a value of 30. The yield went up 30 to 60 to 90. So there might be genetic interactions in here.
But she didn't see it on any high THC cultivars. It was just low THC.
Yes, but to be fair, she only had one high THC cultivar. But the point is there could be some cultivars might benefit from more than others. That's one of the things we want to study is there ones that benefit from more phosphorus. The ones we've tested have not.
Yeah, the test that you have done, you haven't seen an increase in benefit of yield or quality or THC or oil production using phosphorus.
Yes.
But over too much phosphorus really doesn't harm the plant in any way.
Yeah.
But it harms the environment.
It harms the environment.
What is it about phosphorus that harms the environment?
It's the limiting element for algal growth in almost all bodies of water, even in the ocean, lakes, rivers. So if they get high phosphorus, the algae just take off and they turns the water to split pea soup, kills the fish, and it has toxic compounds in the algae that are just hard on other aquatic life. So it's algal blooms are a bad thing environmentally, and they're caused by runoff and leaching from agriculture. Not all of them, but a big portion of it. And to this same topic, academics nationwide are trying to get corn and soybean and wheat growers to put on less phosphorus. You don't need so much. They say, well, it might be a good year. I'm going to put on the phosphorus. So we raise it in our solutions to 30 parts per million.
Which is more than adequate.
Now, you want to make sure you get your customers to and understand that the studies indicate that's very adequate level.
Yeah, if you talk to some of our growers, they would say, "Oh, well, we need to add PK, to your fertilizer line because we think." But even just ProLine as it sits, it has more than adequate, phosphorus. Mm-hmm. But we even have cultivators adding more. Yeah, unfortunately.
Yeah, and it's not just cannabis. If you go up the road, my colleagues at UC Davis trying to get all the fruit growers and the nut growers to put on less phosphorus. And they just say, that something doesn't look right. I'm going to put more fertilizer on. That's a common thing in all of agriculture. And the over-fertilization has detrimental environmental effects.
Do you think, as far as field crops, 100%, but do you think that changes with like indoor greenhouse or indoor facilities?
Well, in principle, no. But in the field, first of all, there's a big volume. I mean, the roots can spread out. They can get phosphorus from a whole bunch of places. If you can imagine a field, it might be a cubic meter of soil, of Rootzone, and then we go down to just a pot, like a Grodin cube. It's a tiny fraction of the field, so we have to keep putting it in. And that's where mass balance comes in. How fast is the plant growing? How much do we need to add to get there? But it's quite different in the field because most of the phosphorus is on the solid phase of the soil. It's in the soil. And the plants, it can be very dilute because the roots spread out like crazy. Yeah.
I know when I was there and I visited you, you were doing a lot of studies on temperature. You had a lot of different chambers that you were running, warm climates and really super cold climates on cannabis. What have you guys been learning about temperature in regards to cannabis cultivation?
Well, first of all, it's understudied because it's hard to study. You need a different chamber for each treatment. But as a general principle, it's good to start warm and cool it off. Now, exactly how warm and exactly how cool, that depends on the light level, depends on the CO2 level. But warmer and in Fahrenheit, I would definitely be in the mid-80s somewhere when you start. Get the plant to develop quicker, it spreads out, it captures the light quicker. And then in early flower growth, the flowers expand quicker too. The downsides, the plant can get taller with warmer temperatures. But then when we cool it off at the end, you stretch out that period of flower fill, and I think you get a higher quality flower because it's maturing in a cooler temperature. You didn't rush it. You let the flower expand by itself. You can get a little more dense bud. And by cooler in Fahrenheit, could be the mid-70s, mid something like that. So that's the principle. The differences from heating and cooling of the Rootzone are typically really small. Just get the shoot optimal and keep the roots the same temperature.
you've seen mid 80s in the beginning of flower up until week 3 or 4, or
Well, in veg too,
in veg.
especially in veg, just to get it to just grow as fast as possible.
So mid 80s veg to early flower. What week are you seeing would be start to cool down?
I would gradually bring it down.
Starting week 2, 3.
Yep, I think that's a good rule of thumb. Maybe even week 4. Taper it, because this is assuming an 8-week crop cycle.
Yep.
One of the other temperature things we've been studying is day-night differential. And for most other crops, we say the day should be warmer than the night. That's just a long-standing thing. It's in all of the textbooks. But that comes from greenhouse cultivation, where it's cooler at night anyway. You're trying to keep it warm at night. And here comes cannabis, 12 hours light, a long 12 hours dark. Should that be cooler than the day? And then the question becomes, could we use a warmer night and a cooler day to make a more compact plant? And this is a concept called DIF, day-night differential. And in floriculture crops, this is widely used. They use a cool day and a warm night. It's really odd, but it makes the plants compact, and it's very effective, and it's widely used in many floriculture crops to keep a compact plant. I'm going to talk in Celsius for a minute, but
That's
take
fine.
the numbers and double them, and you have Fahrenheit. +6 would mean, say, 30-day, 24-night. That would be +6. If you did 27 constant, that would be 0 diff. Same average temperature, just 0 diff. But now you keep going, and you get 30-night, 24-day, and that would be a -6 diff. You're going to negative levels.
Now, with the negative levels, and you're seeing the plant growth get shorter, stunted, right?
And that's what happens in many floriculture species. We have not seen that in cannabis. We've not seen that we can run this negative diff and make the plant more compact.
So it doesn't matter. Negative diff.
Well, it turns out negative diff also reduced yield.
That's why I was going to say it would reduce the yield, because it seems like if the plant
It's backwards.
Yeah, well, not only backwards, but it's not getting warm. We know what happens when you don't have high leaf surface temperature. The plant's not working as hard, and it affects yield and overall growth. So that's probably why you're seeing a negative impact on yield.
Yeah. But I will say a 0 diff, same temperature day and night, that works beautifully. And that's what I often recommend.
So that's big, because growing up in the cannabis industry, going to all the hydro shops and everybody on talking, they're like, yo, you need a 10 degree variant at night versus day. You need to be
10 degrees cooler at night.
10 degrees cooler.
Yep.
But of course, you can get big differences in the middle of summer too. Hot day, cool night. But maybe that helps plants become more reproductive. We have not seen that in cannabis.
So it doesn't matter day-night temperature.
Yeah.
As far as the diff. Diff can be 0.
It could be
and
0.
you won't see any negative or positive impacts. It's just the same.
It's just the same as a warm day, cold night. Yeah. If you were running a greenhouse, you don't want to have to heat it at night. It's expensive.
Yeah.
So you let it cool off just to save money on heat. Exactly. Now we're in indoor agriculture. God, we're cooling all the time. We can have whatever night temperature you want. It doesn't cost hardly any more to have a warm temperature at night indoors. So I think we need to rethink this day-night differential for all crops in indoor agriculture.
Now, in regards to temperature and CO2, are you seeing any benefit of adjusting temperature when you're adding CO2?
Yes. This is well studied, and it comes right from the biochemistry of photosynthesis. And now this is going to sound like a beginning plant physiology class, but that's okay. Maybe we'll inspire listeners to study plant physiology. The name of the enzyme that binds CO2 is called rubisco. That enzyme gets confused, and oxygen sticks to it sometimes. So here's the enzyme. It's supposed to be binding CO2, but it binds oxygen. And when that happens, it's like a car backfiring. It's a boom, boom. It's a waste of energy. If we enrich CO2, the enzyme doesn't backfire anymore. It's got plenty of CO2. It's super efficient. I suppose in a car analogy, it's like using some kind of jet fuel as a gas. It really runs efficiently. This is called photorespiration, when the oxygen binds to the enzyme, and it's worse at high temperatures, much worse. So when we add CO2 and the system's running efficiently, now we can elevate the temperature because the enzyme runs smoothly even at high temperature when we have elevated CO2. So we know from biochemistry studies and multiple studies on photosynthesis in crop plants, the temperature optimum shifts up 3 to 5 degrees Celsius when you have high CO2. The temperature optimum for photosynthesis shifts up.
So when running CO2 on an indoor facility, you're saying that you would possibly see better results in early flower upwards of 90 degrees Fahrenheit?
Oh, no, when I talk about the mid-80s, that's assuming high CO2.
Oh, okay, good.
Yeah,
Okay, I'm glad we clarified that.
Everything we do is elevated CO2.
Okay.
The only time we don't have elevated CO2 is when we do a CO2 stress study, and then we turn it off. But
so in regards to the mid 80s, that's with elevated CO2.
Yeah.
That's the peak. I'm glad we clarified that. Because that weren't running CO2 go in and pump 85 degrees.
It could be a little hot, yep.
Yeah, so no CO2, 78 to 80.
Yeah, in that ballpark.
Yeah, that's about what we usually run. Yeah, in regards to humidity, where do you like to kind of, where do you see it being the optimal?
Yeah, I think there's a lot of things that are underappreciated, like CO2 enrichment. And there's some things that we over worry about. And in some ways, humidity or vapor pressure deficit is something that I've seen people over worry about it. The big deal is to keep it low enough so you keep microbial counts down. You got to be below 60%. You don't want botrytis. You want to keep microbial counts low. That's huge. But plants, in theory, could grow a little bit better with high humidity. Again, they don't have to transpire as much. But when we water them, they just don't have water stress. So the benefit of high humidity is really quite small compared to the risk of getting high microbial counts. So we're always trying to keep the humidity about 60%. And then, like everybody, blow a lot of air so it's 60% in the canopy, not just 60% up here, but 60% down where the leaves and flower buds are. And I think cannabis growers do a real nice job of that.
Yeah.
You see a lot of big fans.
Regarding humidity, where do you see it kind of having a negative effect on cannabis on the low side?
Oh, I think if you got down below 40%.
Below 40.
Yeah, now you're starting to make the plant, it's evaporating a lot more water. Mm-hmm. But typically, the plant's putting so much water in the air, it's hard to get it low. You got to have a lot of dehumidification capability to get it low.
Yeah. No, that's good to know. So between no lower than 40%, and you really don't want any higher than 60%.
Yeah, and I would even say 60% in the canopy.
Yeah, in the canopy. That's important because it could be, 60% in the room, but without good air circulation, you could be 70, 80%. Yeah, I've seen better luck with stationary fans and getting a nice consistent airflow through the room, not oscillating.
Yeah,
I'm not a big fan of oscillating fans.
Yeah. Yeah. No, I'm not either. But they're often used.
Yeah, you're seeing them a lot.
Really, if the one thing we can do keep microbial counts low is blow it with fans in tubes with holes in them into the canopy. So you got your dry air coming and pushing the humid air up.
So for 2023, we're partnering on research and development. That's going to be fun. Yeah,
it is.
We're going to learn a lot. I think you have a formulation, and we want to do a side-by-side at Jungle Boys in our R&D room and go side-by-side your formulation versus our formulation, which, some things are similar, some things aren't. But it would be interesting to see what that looks like. We're doing a side-by-side blended versus pro. So this is kind of the SOP to use fade with blended. Have you done any studies on nitrogen use in the flower phase? Nitrogen levels?
You mean like flushing? Is that what you're thinking? Or
not really flushing, because we're not flushing because we're switching out with calcium chloride and micronutrients. But the effects of lowering nitrogen during the end of the flower cycle.
We have done a study with that, and it was related to precision stress and bring the N down. And in that study, we started to. Now we went to 0 N. We didn't bring it down, but the N just went down in the tissue, and the yield went down as we cut the N. If we only cut it for a week, that wasn't a very big deal, but 2 weeks was bigger, 3 weeks was bigger.
Where were you testing it at? What week did you test the?
Well, it was multiple levels. 1 week, 2 weeks, 3 weeks, and 4 weeks. 4 weeks was really detrimental.
4 weeks into flower? Week 4?
Yeah,
4 weeks.
because there were 8 weeks total. So
yeah.
After, you could say we cut the nitrogen after week 4, after week 5. A lot of stuff we do is to get a curve. I mean, we knew that was going to be detrimental. We just wanted to see how bad it was.
What did you see at week 6? Cutting nitrogen.
It was detrimental
to yield.
Yield.
What did you see in?
Now, having said that, they were never over-fertilized. They were just adequate. They didn't have any reserves going into week 6 either, because our solution is fairly dilute.
Yeah, so you're only feeding 2 EC. Mm-hmm. And we're upwards of 3, sometimes 3.5 with using LEDs. Yeah. So there's a lot more nutrients inside the tissue of the plant.
They can store excess nutrients. There's, as you guys know, there's so many factors here. There's one yield, which you just weigh the flowers. That's easy. How about flower quality? Doing that kind of science is it's just tricky. I mean, we do a study and then we go, is this reproducible? We do it again. We try it with some different ways, to different interactions, and gradually you get some paradigms that you guys do that too. Tricky. Sometimes people do a study and this was 10% bigger.
Yeah,
we're done. That's going to always happen.
No, we're always doing it. We never stop. We didn't see a significant impact in yield, but in our test. But like Bruce said, we're feeding a much higher start EC than he was.
And have you found keeping calcium high to the end helps minimize botrytis?
Yeah, that's big.
That's one element that's.
What did you see in your studies on eliminating nitrogen on finishing the plant? Did you see an increase of increase of
cannabinoids?
Yeah.
Mostly our strains are high CBD, but they still have THC. No, we didn't see an effect on it. There was just no difference. You would think they might concentrate a little. That's the reasonable hypothesis. But on the other hand, cannabinoids are always turning over. They're synthesized and degraded, and maybe not as fast as some other things. So if there's less photosynthesis with lower nitrogen, there could be less synthesis of cannabinoids too. So it's a tricky balance.
Did you
down the
see a change in the flower structure or color?
Yeah, no, we didn't do rigorous studies on that, but there wasn't a
you didn't
visual.
do. Okay.
Yeah, one of the things we have started to look at is just bud density, and just can we get some nice dense buds as opposed to airy buds?
Yeah, we saw a significant change in flower color in some cultivars. You know, it was significant. More cultivars were turning purple for some reason by pulling that nitrogen.
Yeah, I could believe that. I mean, that's anthocyanins, and if there's a little stress, the anthocyanins can go up.
Yeah, that's what Chris was saying. Stressing we're causing a little bit of stress at the end, which is changing the plant. So
anthocyanins are in red lettuce, and there's all various amounts of red lettuce, and we and other people, you grow lettuce really optimally, and it's not very red.
No, it's going to be fun this year to work with you on a bunch of different studies, and we're really excited about it. For us, I think it's going to be big to just cut through the misinformation on plant fertilizer in the cannabis industry. It's going to be nice to have your help to just kind of get to the truth, right? And that's what we're big on. And I think, obviously,
Let me mention one key thing about differences in approaches. Because we're a research lab, we work with whole plants and we work with whole communities of plants. But a whole community of plants might be 4 to 6 plants for us. It might be half of this table is our experimental plot. And we have to be real careful. If the plants flop over on the side, that's going to make yield go up. Sometimes we put a fence around them to hold them in so we can extrapolate to a bigger community of plants. But it can be tricky to do the research on a small scale and extrapolate to a big scale. And that's where you come in, doing it on a big scale. Now, does it translate?
No, cool, Bruce. I appreciate you making the trip out, and really value your time and value the partnership, and looking forward to 2023. So thank you.
As they say in the Olympic model, Citius, Altius, Fortius. And that means when you look that up in Google, it means faster, higher, stronger. So I like to use Citius, Altius, Fortius.
Nice. It was a pleasure. Thanks, Bruce.
Thanks.